YouTube. I do YouTube.
That sounds like that we did. Okay. So we talked about DNA replication in prokaryotes and eukaryotes. Now we're going to talk about transcription in prokaryotes and eukaryotes. Remember, 70% of your exam is on this molecular biology stuff. So once again, if you see history stuff, just skip it. How many RNA polymerases are there in prokaryotes?
RNA polymerase.
In prokaryotes, there's only one.
One.
How many in eukaryotes?
3.
In prokaryotes, the RNA polymerase is made of what components? Alpha, beta, beta prime, sigma,
That's when I hold my hand. It wasn't on the test.
oh, technically it's 2 alpha, so it's 3 types of subunits plus delta, but a total of 4 subunits plus delta, plus sigma. I keep saying delta, plus sigma. Your sigma subunit is not part of your core polymerase. Your sigma subunit is separate, and when it adds to the core the core RNA polymerase put together, that is called what? A hollow enzyme. Very good. Your sigma subunit is important because that's the one that recognizes a promoter sequence. So we know there's several types of promoter sequences. In prokaryotes and eukaryotes, you hear some terms. You hear the term Pribnow box. And which one is that found in? Prokaryotes or eukaryotes?
Prokaryotes.
Prokaryotes. The other one, the top box there for us is eukaryotes. Okay.
Isn't the Tata box like, it was like downstream or something like that?
Yep, that one can be found downstream in eukaryotes. To talk about prokaryotes, there are very similar stages in transcription as to translation. There's an initiation phase, there's an elongation phase, and there's a termination phase. In prokaryotes, in initiation, you need to have the RNA polymerase, you need to have the mRNA, you need to have the ribonucleotides, and you need to have the promoter sequence. Okay? So initially, you see terms called the closed promoter sequence and the open promoter complex, the closed promoter complex and the open promoter complex. Initially, the DNA is closed, and then when you start transcription, it is open. Which one do you think the RNA polymerase is more strongly attached to, the closed or the open one?
The open.
The open one, because once you're doing transcription, RNA polymerase is tightly bound to the open one. If it is not open yet, RNA polymerase may dissociate. So it is more strongly bound to the open complex. Let's see. Mm-hmm.
What's the relationship between the promoter recessive sequence and RNA polymerase?
So there's 2 promoter sequences in prokaryotes. Oh, so when you use the term consensus sequence, what does that mean? So they use that term, so let's see if I can find an example of it. It's like for the Pribnow box, it's like like TA, it's some pattern. That's what the consensus sequence means. It's a specific pattern that is repeated on the promoter sequences. Oh no, that is the promoter sequence. The consensus sequence is the promoter sequence. It's not upstream of the promoter. The promoter is upstream of the transcription site. What they mean is if a promoter sequence is closest to the consensus sequence, that means there will be the RNA polymerase will bind more strongly to it. So you can think of it that there's different types of promoters. They're all going to be either very similar to the consensus sequence or a little bit different. The ones that are more similar to the consensus sequence will bind stronger to the RNA polymerase for transcription. Yeah, it's close. It's similar to the consensus sequence, yes. And that's during initiation. So initiation RNA polymerase bonds to the promoter sequence, and we go under initiation. It bonds to the, you need to know where the Pribnow box is and where the unnamed one is, which is minus 35, I believe. I don't think you'll have to identify them, just know that where each one is. So you won't be given a sequence and asked where is your promoter sequence going to be, where is your transcription going to start. Let's see. By the way, I'll resend those video links concerning transcription and translation. We already went over that. Okay. Talking about chain elongation. It is pretty simple. It's much simpler than the elongation in translation. All it is bringing in ribonucleotides one at a time, and attaching it to the 3' end of the nucleotide in front of it. Remember that we generate DNA in the 5' to 3' direction. We transcribe mRNA in the 5' to 3' direction. And we do translation by reading the mRNA in the 5' to 3' direction. It's all about 5' to 3'. So because we're going in the 5' to 3' direction, your nucleotide keeps adding to the 3' OH end of the front nucleotide. Oh, you may get a question to ask which of these enzymes do not have repair activity, or which ones do have repair activity. We remember that in DNA replication, DNA polymerase 1 and 3 both have repair activity due to what specific exonuclease? 3' exonuclease. We know that the clean-out fragment is made out of a certain fraction of DNA polymerase 1, which includes the polymerase and the 3' exonuclease. Therefore, the clean-out fragment also has a repair activity. If on your multiple choice he includes RNA polymerase, does that have repair activity?
No.
No, there is no function. There's no way for RNA polymerase to edit mistakes. However, it is still fairly accurate. It doesn't mean it's making errors all the time. You just need to know it does not have an editing function. You don't have to worry about how fast it generates the mRNA molecule. However, you may need to know in general, if it runs into a sequence that has a lot of G's and C's, do you think it's going to be transcribing fast or slow?
Slower.
Slow, because the GCs have 3 hydrogen bonds. It's harder to break the GC bonds. That's why transcription is slower in highly GC-rich regions. We know in DNA replication, because you're unwinding DNA to replicate DNA, in transcription you're also unwinding DNA to transcribe it, so you must also have topoisomerases relieving supercoiling, just like in DNA replication. Prokaryotic termination, you need to know the 2 ways that termination happens in prokaryotes. There's something called row-dependent and row-independent. For row-dependent termination, that means there is a protein involved called the row protein. That protein looks for the RNA polymerase complex, binds to it, and causes everything to dissociate from the DNA. So that one's a pretty easy one to memorize. The other one is the row-independent termination. This one, it does not involve any proteins. It is only dependent on the sequence itself. And what feature of the what type of characteristic would you describe the DNA that will form the row-independent termination sequence? Hairpin loops. How about inverted repeats, right? You may be asked, both are correct. You may be asked a question, given a DNA sequence, or given several DNA sequences, which one is going to form a hairpin loop, or which one is going to be where transcription is terminated. So you need to be able to identify inverted repeats of some DNA sequences that he gives. Yeah, you need to find out which one has inverted repeats. I believe there was a practice question on that. So yeah, make sure you're able to do that. Let's see. So the hairpin loop, when the RNA polymerase generates the mRNA and it forms a hairpin loop, the RNA polymerase doesn't like following that path, so it dissociates from the DNA. There's more specifics on it, but you don't need to know the highly detailed stuff. This experimental stuff, let me check real quick. Yeah, you don't need to know about GFP, the green fluorescent protein. You don't need to know anything about Okay. For transcription regulation, he talked about positive and negative control, induction and repression, right? Negative, so if you see the term negative induction or positive induction, the final result is always inducing transcription. The terms negative control and positive control tells you if an activator is involved, inducer is involved or a repressor is involved. So if you were told that some operon is negatively induced, you know that we are turning on transcription by removing a repressor. Okay? So you may be asked, what is used to activate transcription in negative control? And you'll say co-inducers bind to the repressor to make it dissociate from the DNA strand. I'll give you a practice question that's like that. Positive control we know is involving inducers. So positive induction means you must have a co-inducer bind to the standard inducer that allows it to bond to the DNA to allow transcription. So you need to know both types. He's given details on the lac operon many times. He's asked questions on the lac operon, so I'm sure you guys are very familiar with that. I'll send you the video link on that again. Remember, he's not going to give you extra credit questions on your final. So if he asks about the lac operon, it'll be a multiple choice form this time. Let's see.
Josh, when you said that there wasn't going to be any structural things for the first part.
Yeah, like amino acid. Yeah, like in other words, he won't ask you to identify which of these is like lysine or something, which of these is maltose or something. Yeah, he won't ask you those types of structure type questions. He only cares about big picture for exam 1 and 2, so you don't have to worry about the structures. No, so like the sphingosine, all those things, you may need to know how to classify them, but you will not need to be you will not be asked to identify whether a structure is like cerebroside or something like that. You need to know that a sphingomyelin is like sphingosines, cerebrosides, ceramides, gangliosides, and there's probably one more. These are terms that you will find in your exam 2 notes. You will not be asked to identify the difference between them, but just know how to classify them, I should say. Let's see. Once again, you don't need to know anything about the tryptophan operon, so you can skip that. So he doesn't care about specifics about transcription factors, but you need to know that transcription factors are required in eukaryotes. I won't go over, I'll go over briefly the part 2 of this PowerPoint, but in terms of eukaryotic transcription, he only wants you to know what certain terms mean, what like enhancer means, what response elements means, what promoters mean, things like that. So you just need to know the definitions of those key vocabulary terms. You'll see those in the first few slides in a little bit. Other things with eukaryotic transcription, you need to know about the only structural motif you need to know is the zinc finger, right? Yeah, we'll go over that as well. I'm guessing that's not in this slide. Yeah, I'll go to that. We know that in eukaryotes we have chromosome structures. We have chromatin that's wrapped around, I think this was in chapter 11, I believe, where they talk about nucleosomes. What are nucleosomes? Those are small little protein structures where DNA is wrapped around so that you can compact DNA in eukaryotes. So if you're doing transcription in eukaryotes, Not only must you open DNA, but you must also open the nucleosome to access that DNA. So these are some small differences between eukaryotic and prokaryotic transcription that you need to know. You need to know what promoters are, enhancers are, response elements, and silencers are. Oh, and you need to know which polymerase is the one involved in transcribing mRNA, and that's RNA polymerase 2. In terms of small details, RNA polymerase 1 is the one that does rRNA, and RNA polymerase 3 is the one that does tRNA. I don't know if they say that here, but that is somewhere in the notes. So once again, here's this very important slide that defines all the important terms in this chapter, in this part of the chapter. I'll let you guys do that. You need to know the roles of transcription factors. You do not need to know if transcription factor 1 does something, transcription factor 2 does something, just know the general roles of each.
That's the one that he Pacey's added that to last week.
In terms of when they're talking about RNA polymerases, the previous SI leader mentioned that sometimes he asked questions concerning the yeast polymerase. So I would suggest you look at this slide for that. But again, if the TA mentions something on Saturday, go by what she says. I'm going sort of by past history on this part. Let's see, I gotta skip this. Here's the Tata box. We mentioned the Privna boxes in prokaryotes, the Tata boxes in eukaryotes.
There's a Tata box at the library.
Okay. From here on out, I would not focus my studying on anything past this part. Okay. So for the final, I would not focus on anything past this part, except for structural motifs, you know what I'm going to mention, the zinc finger, wherever that is. Here it is. You need to know everything about the zinc finger. You need to know that there's 2 classes. You need to know that it prefers to bind to the major groove of DNA. So in DNA, in that double-stranded sequence, there is minor grooves and major grooves. Structural motifs focus on the major grooves. So you need to know everything about the zinc finger. Here are the 2 structures, C2H2 and CX. You need to know everything here. Oh, I guess they don't care. All right, so everything else.
So no capping and methylation?
Oh, in the splicing part, you only need to know the basics of it, yes. You need to know that when you generate your eukaryotic mRNA, you must remove the introns, you must add a G prime cap, a 5' G cap, and a 3' poly A tail. So those are the editing you must do to eukaryotic mRNA. Pre-mRNA is also known as HN mRNA, heterogeneous mRNA, so just in case he uses that instead of pre-RNA, they are the same thing. HN mRNA and pre-mRNA are the same thing. And I'm assuming, yeah, that's post-transcriptional processing, so those 3 basic steps that you have. And you don't need to know anything about splicing, so the rest of this you can skip.
What about the Hox or hoax?
The Hox genes in the homeobox. I think you only need to know the definition of it. You don't need to know these.
Okay.
Yeah, he gives you like 5 slides on it. You
Yeah.
don't have to know for the final. You don't have to know.
What's the definition?
Yeah, it's on here. Yeah, and once again, remember that he may alter the final a little bit. I don't think he's going to alter it tremendously, but if your TA mentions something on Saturday, make sure you take note of it.
Can you make a note
we're
that
not
you
allowed
should
to
make
take
it
notes.
easier for us?
If people are wondering how the curve is going to be on the final, since it's mostly exam 3, the curve will probably be similar to on the final to exam 3. No, it's just
A lot of people fail exam. Yeah. You don't need 7 extra credit points.
Does anyone remember how many questions it is?
110.
10 questions would be great. Yeah, I think it's
What?
It's 100. It's 100.
What'd you say?
How does he make so many questions?
What did he say?
So,
I don't know. 10 will be graded or something?
if you've gone to his office to review previous exams, you may see that the final will look very similar to the previous exams. He will take some questions from the exams you've taken. He will take some questions from the practice exams. He may add a few that you haven't seen. He may just edit a question you've seen before and ask maybe for the opposite thing. He won't really come up with brand new questions you've never seen before. It'll be stuff you've seen before or can infer from a previous question. So if you're going over your practice questions, don't memorize the answers per se. Make sure you know why they picked an answer there so that you can, in case he changes the question a little bit, so you still get the correct answer. So here's some more. These questions that I have here will cover from chapter 11 to transcription to 29. So, true or false? We gotta go through true or false for each one of these. Template strands are only copied continuously. This is a little bit tricky.
False.
Not template, yeah, this is false. Not template strands. The leading strand is generated continuously. But does that mean the lagging strand isn't a template strand? The lagging strand is also a template strand, so A is incorrect. Okay. And we know the lagging strand is generated discontinuously, non-continuously. In E. coli, the replication fork copies DNA continuously and segmented in a 360-degree direction. This is phrased a little bit differently than you will see on an exam, but remember that prokaryotic DNA is what shape? Circular. So it is generated well, it's not technically 360 degrees, so I should have put 180 degrees here, but remember that you have one origin of replication, so DNA is generating in one half of the circle, DNA is also being generated in the other half of the circle simultaneously, so I should have put 180 degrees there. What's that? Yeah, bidirectional, that's what I'm looking for. So let me edit B, but it should be true. DNA is unwound by gyrates without breaking it. That's false. It's not gyrates that does that. Or helicase is the one that opens it without breaking it. Gyrates is doing what ahead of the replication fork? Relieving supercoiling by breaking the DNA. So we know that one is incorrect, kind of two ways. Eukaryotic DNA can contain more than 2 replication forks.
True.
That is true. We remember that prokaryotes only have one origin of replication. Eukaryotes must be able to replicate a bunch of DNA in a short amount of time, so they must have a bunch of replication forks. Replication occurs mainly during G2 and G1 phases when cell growth occurs. So this is a biology question. That's false. In what phase?
S phase.
S phase. Very good. Fill in the blanks. Bidirectional replication starts at the what site? So I mentioned this term a lot. At what site does DNA replication start at?
Origin.
Origin of replication, also known as ORI-C. O-R-I, capital C. A blank is bound to deter locus to prevent helicase activity. What protein is that?
TUS.
Very good.
I found that other day.
High blank means a high number of successive cycles of nucleotide addition.
Processivity.
Processivity, very good. High processivity means a high number of successive cycles. And what feature helps DNA polymerase 3 have high processivity? The beta clamp, very good.
Oh, beta clamp.
A primer needed for replication is formed by what enzyme? Primase. The process in which an RNA template is transcribed to complementary DNA is executed by what enzyme?
Reverse transcription.
Reverse transcriptase, very good. Which of the following is true replication? It creates a complementary strand relying upon base stacking interactions. If I were to phrase it a little bit better, it would be base-to-base interactions. Is that correct? You're generating a complementary strand. How do you know what nucleotides to bond to A? You know due to hydrogen bonding, you know, due to base interaction, base-to-base interaction. So A is correct. Usually, one replication fork must move 360 degrees around the entire chromosome in E. coli, so we know that is false. It only needs to move, one fork moves in one half, the other fork moves in the other half. DNA polymerase 1 adds a new nucleotide to the 5' end of the existing DNA molecule. That's false. All DNA polymerases only can add on the 3' end. That's why the lagging strand is discontinuous. DNA polymerase 1 adds a new nucleotide in the 3' end of the template strand. That's a little bit, it's a bit awkwardly phrased. It is false because it's not adding to the template strand, it's adding to the 3' end of the primer. So if we're talking about, or to even make it more broad, it's adding to the 3' OH end of the nucleotide in front of it. So that's what you're continuously adding on. Why does DNA polymerase 3 need a primer? Because DNA polymerase 3 can only add nucleotides to a 3' end of something. That's what the primer provides, is a 3' end for the DNA polymerase to add nucleotides to. So DNA polymerase 3, we'll move on to the next one. DNA polymerase 3 can also synthesize Okazaki fragments. So I mentioned that this wasn't really mentioned in your notes, but it is true that DNA polymerase 3 can synthesize Okazaki fragments. But which one is the one that removes the primer and replaces the primer with DNA? DNA polymerase 1 does that. So DNA polymerase 3 does synthesize Okazaki fragments. DNA polymerase 1 is the one that removes the primer and replaces the primer with DNA.
Exonuclease activity, right? Is that because it has exonuclease, right?
What's that?
Is it because DNA polymerase has exonuclease?
DNA polymerase 1 has the correct exonuclease to remove primers. Which exonuclease removes primers? 5'. DNA polymerase 3 does not have that. DNA polymerase 3 functions mainly in DNA repair? So, mainly, no. Its primary function is to generate DNA. Its secondary function is for DNA repair. DNA polymerase 3 polymerizes DNA in the 5' to 3' direction. Oh, and when I say DNA repair, I don't mean it searches for errors all throughout DNA. Where is it immediately only looking for errors?
Transcribed.
Only in the DNA it just made. So that, and which activity is the repair one? The 3' one is the repair activity. Very good. So for C, DNA polymerase 3 polymerizes DNA in the 5' to 3' direction. That's true, so the answer is A and C. Okay, talking about reverse transcriptase. Can reverse transcriptase be a telomerase that uses its own RNA as template? Yes, and that is what a telomerase is. It creates, it extends DNA sequences by adding RNA nucleotides, and then it does reverse transcription to replicate DNA. Oh, did it skip? Yeah.
Number 5. You missed
Okay,
one.
yes. All right, well, we'll do 6 first, then we'll do 5. So 6, reverse transcriptase is contained in all RNA tumor viruses. That is true. Reverse transcriptase allows the transcription of complementary DNA from an RNA template to a DNA-RNA hybrid. That's true, that's the first activity. The second activity of reverse transcriptase is to hydrolyze that hybrid. And the third activity is DNA-directed DNA polymerase. Reverse transcriptase can be inhibited by HIV therapy. That is true as well, so all of the above. Number 5, which of the following statements is not true? Replication occurs during the S phase of the eukaryotic cell cycle. That's true. Helicase is an ATP-dependent protein that unwinds DNA during replication. That is true. So a helicase, make sure you know, is ATP dependent. And does it break DNA strands when it unwinds?
No.
DNA polymerase 1 uses its 3' to 5' exonuclease activity to remove primers.
No.
Which
one breaks it? Which one breaks it again?
Primers. The 3' does what?
They're proofreading.
Repair/edit, fixes mistakes. Ligase seals nicks in the DNA by covalently linking Okazaki fragments.
True.
That's true. DNA polymerase 1 has 2 types of exonuclease activities. That is true as well.
True.
Okay. Fill in the blanks. In negative control, meaning a repressor is involved, there are repressor is deactivated by what binding to it? A co-inducer. Blank activates repressors in the negative control of the tryptophan operant. Oh, I forgot to remove that, don't worry about it. So skip B. When glucose is present, blank concentration diminishes, preventing the binding of CAP. Remember that the answer is CAMP. Remember that CAP does not want glucose. CAP wants a low glucose concentration because if it has a high glucose concentration or any amount of glucose is present, the cyclic AMP concentration will decrease, preventing the binding of the inducer for the CAP protein. In eukaryotes, RNA polymerase what transcribes mRNA? So the choices would be 1, 2, or 3. 2, and then what transcribes tRNA? 3, and 1 transcribes rRNA. Blank allows eukaryotic RNA polymerases to interact with their promoters.
I think I wrote that on my hand.
I think I should have prokaryotic.
Oh, yeah.
Okay. So this one is just transcription factors. Don't worry about what transcription factors. Just know that transcription factors allow eukaryotic RNA polymerases to interact with their promoters. Okay.
I can start a YouTube channel.
Is it?
How does RNA polymerase differ from DNA polymerase? So we talked about a lot of these. How about RNA polymerase reads the sense strand, DNA polymerase reads the sense strand. So this one has to do with sense strand or anti-sense strand. Which one is your template? It's the sort of the opposite. Your anti-sense strand is your template. So they do not read the sense strand. RNA polymerase polymerizes in the 3' to 5' direction, creating an anti-parallel and complementary strand. Almost correct. Polymerizes in the 3' to 5' direction? No, that's not correct. RNA polymerase does not require a primer. DNA polymerase requires a primer.
True.
That is true. RNA polymerase uses both DNA strands as a template. DNA polymerase uses only one template strand. So that's false. And when it's phrased like this, they mean that DNA polymerase can use both strands simultaneously, while RNA polymerase, it won't pick just say like the top strand and never use the bottom strand, but it will only use the top strand for one protein as the template. And then if it wants another protein, it may use the bottom strand for that. So the RNA polymerase only uses one strand at a time for template. DNA polymerase can have a leading strand and a lagging strand, so it can use both as a template. So the answer is C. Which of the following is true regarding transcription regulation in eukaryotes? It only includes promoters and enhancers.